Macrophages are scavenger cells that can be induced by pathogens to present foreign antigens to naive T cells. Many of the microorganisms that enter the body are engulfed and destroyed by phagocytes, which provide an innate antigen nonspecific first line of defense against infection. Microorganisms that are destroyed by phagocytes without additional help from T cells do not cause disease and do not require an adaptive immune response. Pathogens, by definition, have developed mechanisms to avoid elimination by innate immunity, and the targeting and removal of such pathogens is the function of the adaptive immune response. Mononuclear phagocytes, or macrophages, that have bound and ingested microorganisms but have failed to destroy them contribute to the adaptive immune response by acting as antigen-presenting cells. The adaptive immune response is in turn able to stimulate the microbicidal and phagocytic capacities of these cells. Resting macrophages have few or no MHC class II molecules on their surface and do not express B7 molecules. The expression of both MHC class II and B7 molecules is induced by the ingestion of microorganisms and recognition of their foreign molecular patterns. Macrophages, like tissue dendritic cells, have a variety of receptors that recognize microbial surface components, including the mannose receptor, the scavenger receptor, complement receptors, and several toll-like receptors. These receptors function in the innate immune defense mediated by macrophages. They are involved in the uptake of microorganisms by phagocytosis and in signaling for the secretion of pro-inflammatory cytokines that recruit and activate more phagocytes. In addition, they can play the same role as tissue dendritic cells and allow the macrophage to function as an antigen-presenting cell. Once bound, microorganisms are engulfed and degraded in the endosomes and lysosomes, generating peptides that can be presented by MHC class II molecules. At the same time, the receptors recognizing these microorganisms transmit a signal that leads to expression of MHC class II molecules and B7 molecules. Thus, the induction of co-stimulatory activity by common microbial constituents occurs in both dendritic cells and macrophages. This is believed to allow the immune system to distinguish antigens borne by infectious agents from antigens associated with innocuous proteins, including self-proteins. Indeed, many foreign proteins do not induce an immune response when injected on their own, presumably because they fail to induce co-stimulatory activity in antigen-presenting cells. When such protein antigens are mixed with bacteria, however, they become immunogenic because the bacteria induce the essential co-stimulatory activity in cells that ingest the protein. Bacteria used in this way are known as adjuvants. Self-tissue proteins mixed with bacterial adjuvants can induce autoimmune diseases, illustrating the crucial importance of the regulation of co-stimulatory activity and discrimination of self from non-self. And that's macrophages are scavenger cells that can be induced by pathogens to present foreign antigens to naive T cells. Have a great day.